So with osmosis and diffusion, there are a couple kind of major ways that we can think about the way that water is going to move. So there are a couple of kind of different scenarios that we could find with some of our different solutions. The first one is called a hypotonic solution. A hypotonic solution is one that has very low levels of solute. So it's very pure. It does not have very much stuff dissolved in there. So, for example, if we take a cell that has 90% water and 10% solute, stuff like salt and sugar and proteins and stuff like that, and place it into a cup that's 100% pure water, is the water going to go into the cell, or is the water going to leave the cell and go into the cup? So our cell is 90% water, and our solution is 100% water. So where is the water going to go?
In the cell.
Exactly. Estrella and Victoria both agree with you. So yeah, we have a high concentration of water outside, a little bit lower inside. So water is naturally going to go into the cell. This is not great for the cell because cells can only fit so much water inside of them. And if they get too much water inside of them, they could pop like an overinflated water balloon. So hypotonic solutions are where you have a lower amount of solute. So essentially means you have a really high water concentration. So if a blood cell is placed into a very hypotonic solution, the water is going to go into the cell and cause it to potentially burst. So hypotonic means lower solute, which is another way of saying more water. So just like with our salt example earlier, the beaker of pure water was a hypotonic solution compared to the bag of salt water that we plopped in there. So when you see, we're going to talk about a lot of tonic, blank tonic solutions. So when you see hypo, I want you to think low solute. So low salt, low sugar, whatever it might be. So hypo is low solute. So if a cell is put in a hypotonic solution, water is going to go into the cell. So that is our first kind of type of solution that we could have. There's also a hypertonic solution, which is where you have a lot of solute. You have a very concentrated solute amount. So imagine we take that same cell that's 90% water, 10% solute, and we put it into a solution of 50% water, 50% solute. So we have like a 50-50 mix of water and like salts and sugars and proteins and stuff. So the cell is 90% water. The solution is 50% water. So where is the water going to go? Is it going to leave the cell or is it going to go into the cell? So if we have 90% water in the cell, and we have 50% water outside the cell, is the water overall going to move into the cell, or is the water going to move out of the cell?
Into the cell.
So the water is going to go from the 50% solution to the 90% cell.
No.
Oh,
It should be the
wait.
90% to the 50.
Hey, there you go.
Okay.
So this is water going from the high concentration to the low concentration. In this case, our high concentration is inside the cell, right? We have 90% water inside the cell. Our low concentration is outside the cell. We only have 50% water outside the cell. So the water is going to go from the 90% out to the 50%. So the cell is going to lose water in this situation. It's going to become shriveled up, essentially. Like when you take a grape and you dry it and turn it into a raisin, same basic idea is going to happen to this cell. So a hypertonic solution is where we have a lot of solute, but not very much water. So in the case of our cell here, water is going to leave the cell and go into our solution out here. So hyper is like lots of solute, lots of stuff, but low amount of water. So hypertonic solutions have a very high solute concentration, which means that they have a lower amount of water, right? Instead of being 100% or 90% water, now they're 50% water, because the other 50% is taken up by our solutes. So if a cell is placed in a hypertonic solution, the water will diffuse out of the cell and into the solution. It'll leave the cell and go out into the surrounding hypertonic solution. So that's what causes these red blood cells, in this case that we're looking at, to become all wrinkled and shriveled up like a raisin, because all the water is leaving the cell and going out into the solution that it's being placed in. So hypotonic is low solute, so it has a lot of pure water. So the water will move from the high level solution into the cell, causing it maybe to swell and pop. The hyper solutions mean we have lots of solutes. So the water is going to leave the cell and go to the solution. It's going to exit the cell to try and balance out with that high solute concentration. So we have one final type of solution to talk about, and that is what's called an isotonic solution. So this is a solution that has the same or extremely similar concentrations to your cell or to the thing you're comparing it to. So if we took a cell that's approximately 90% water and 10% solute and dropped it into a solution of approximately 90% water and 10% solute, what is going to happen? What's the water going to do?
Stay there?
Stay there. Bingo. The outside and inside solutions are basically the same, so the water is already equally distributed, right? It's already balanced. So it's not going to move a whole lot in or out because it doesn't need to. Osmosis says that if our water concentrations are the same, water is not going to move. It only moves if there is a high concentration and a low concentration area. Otherwise, it's just going to stay the same. So that's what isotonic solutions are all about. Iso, like isolated, like alone. No movement between the two. So when the concentrations are the same inside and outside of the membrane. So we are at equilibrium. Everything is balanced on the inside and outside. So if water leaves the cell, water is going to go back into the cell at kind of the same rate. So there's always going to be the same overall amount of water outside and inside this cell. We're not going to have any changes of the cell swelling up and getting big or drying out and becoming all shriveled up because the water is going to stay the same for the most part. So that is what isotonic solutions are referring to. When we have equal concentrations on the outside and on the inside of our membrane. All right, any questions so far about our hypertonic, hypotonic, or isotonic solutions?
No.
All righty. So, quick little quiz to kind of wrap up the passive transport section of our notes here. True or false? Passive transport requires energy to work.
False.
False. It does not. Passive transport happens naturally without needing any energy. Osmosis and diffusion do not require energy. True or false?
False. No,
True.
true.
Ah, true.
Yes.
So these are different types of passive transport. So again, they happen completely automatically and naturally. We don't need any extra energy in order for these guys to happen. It's all natural. All right. This one's a little bit tricky, so think about it for a second. If a cell has 30% solute, so like salts and sugars, and the outside, the extracellular solution, so the stuff that it's placed in, has 50% solute, 50% salt and sugar, is water going to move into the cell or out of the cell? I'll give you a second. Think about what your water concentration is going to be and how the water is going to move.
So I think out of this.
Nasima, you say out?
Yes, I think.
I think
All right, Victoria says
B.
B also out. All right. Anybody else? Any other thoughts or input, what you think is going to happen. If you have a 30% solute cell, put in a 50% solute solution. Is the water going to go in or out of the cell? Straya says D. I just realized that B and D are the same thing. Anyways. All right, so out is correct. So if a cell has 30% solute, that means that the cell is 70% water. And then if outside the cell is 50% solute, that means that it is 50% water. So the cell is 70%, the outside is 50%. Osmosis says we move from high to low. So we're going to move from the inside of the cell to the outside of the cell. So the water is going to go out of the cell and move into the extracellular solution. So the kind of second part of these questions, so obviously we had 3 outs over here. The second part of this is this outside solution a hypertonic solution, or is the outside solution a hypotonic solution?
Hypo.
We got some hypo guesses. Any other guesses?
Maybe hype.
So in actuality, the most correct answer out of all these, oops, hold on, there we go, is actually A. So the outside is hypertonic, because hyper means more solute. The outside is 50%, the inside is 30%. So the extracellular solution, the outside solution, is hypertonic. It has more solute. So because of that, that means it has less water, which is why the water is going to move out of the cell and into the hypertonic solution. The cell is naturally going to lose water if it's put in this situation. All right, well, so good. Thank you for thinking that through with me. As a whole bunch of kind of some of the different stuff we were talking about today, all put together in one question. I'm about it. So water moves from a high to a low concentration if it's 70% inside the cell and 50% outside the cell. It's going to move from in to out. The more solute that you have, the less water that you have. So the higher your percentage of solute, like salt and sugar, the lower percentage of water, right? If you have 10% solute, that means you could have 90% water. If you bump that up to 50% solute, that means you can only have 50% water. And then finally, water goes from a hypotonic, so it'll leave a hypotonic solution and go into a hypertonic solution until eventually, if it's allowed to completely equalize, it'll produce an equilibrium and the solution will become isotonic, where everything is all balanced. All right. Any final major osmosis questions, comments, concerns, things that you're still a little sketchy on when it comes to moving water and molecules passively?
Mr. Mark, I have just a question. Just about the hypo and the hypertonic. What's the difference between those two?
So hypotonic means that you have a low amount of solute, like salts and sugars, and a high amount of water. So hypotonic means like a high water, like it's very, very diluted, whatever you have mixed in there. It's mostly water. Hypertonic means that you have a lot of solute and less water. So whatever sort of solution you have, it's a very concentrated solution. There's not as much water because there's more stuff dissolved in the water. So hypotonic, more water, less solute. Hypertonic, more solute, less water.
Okay, thank you.
No problem. All right. So that is actually where we'll kind of wrap it up tonight. So that was passive movement, kind of in a very general concept, talking about basic flows of water and diffusion of particles. So on Thursday, we'll get into how cells move things passively across their membrane. And we'll also talk about active transport, how energy is used to move stuff across membranes. So I realized about halfway through our presentation that I forgot to start recording. So I'm going to do a quick review of kind of the first couple or the first several slides that we went through, in case you want to come back and watch me describe things again. Plus, it'll be a nice chance for us to kind of review all this stuff before we end today. So today we were talking about cell transport, how cells move things in and out of the cell across their membrane. All cells have a membrane, and stuff has to go across it in order for the cell to stay alive, right? We need food and water to go in, and we need waste to go out. That is what helps the cell stay at homeostasis, basically stay at a healthy level of functioning. It keeps all of its nutrients and pH and water balanced by moving stuff in and out of the cell across the membrane. A big thing that we talked about was solutions, where we have a solute and a solvent. So solutes are things like salts and sugars and proteins and amino acids and other things that are floating around in cells. And water, for the most part, is our solvent. It's what all of our dissolved stuff is floating in. So the more solute that you have in solution, the less water that you'll have. Here's our cell membrane that everything is going to be going across. We have this lipid bilayer right here, 2 layers of lipid macromolecules that keep stuff from going in and out of the cell. And inside of those, we have all these different types of membrane proteins that we'll talk about on Thursday that help cells move the right types of stuff in and out of the cells. So it's a bilayer because there's 2 layers of lipids, and they're kind of studded all across with these proteins. One of the first big concepts that we talked about was permeability, which is how likely a type of membrane is to let certain particles across. The more permeable a membrane, the easier it is for stuff to get through. The cell membrane is only semi-permeable. Only certain things can go through the cell membrane. It's kind of like our little example here, membrane. And this one, it's studded with all these little circular holes. So these big square particles cannot get through. They are blocked from crossing the membrane. But these little circle particles are able to pass. This membrane is permeable to these little dots right here. Passive transport is the main thing that we focused on today. It's where molecules don't need any energy in order to cross a membrane. And it's all about moving from high concentration, where you have a lot of a specific type of molecule, to low concentration, where you do not have as much. Diffusion is kind of the super basic version of this, where you have things like scents diffusing throughout the air, or dyes or particles diffusing throughout a liquid. But again, all about moving from high to low concentration. That movement is due to these particles that are constantly bouncing around and bouncing off of each other. That's what causes them to start off bunched up in these really high concentration areas, but kind of bounce off each other like bumper cars and end up moving to the low concentration areas. And they'll keep moving around like that until they're all totally evenly and equally spaced throughout the mixture. So they're trying to become equally distributed. They're naturally going to become equally spaced apart from each other. Until they do that, they're going to move from the high to the low concentration areas. So in this example that we talked about, we have 2 dot molecules on the left and 6 on the right. And so the dots will naturally flow from the right to the left across this membrane, because there's a higher concentration over here and a lower concentration over here. The squares would go the other way, except they can't cross the membrane. They're blocked from passing it. So even though naturally they want to diffuse to the right, they can't. The membrane won't let them pass. So osmosis is a particular kind of movement where it's specifically water. So osmosis is when we're talking about water passing across a membrane, not a solvent like salt or sugar or something like that. We're still going from high to low concentration though, always going from more concentrated to less concentrated areas. So if we drop a saltwater bag in this pure water beaker, we have a lot of salt and 70% water in the bag, but 100% water in the beaker. So the water will naturally move into the bag. There's 100% in the beaker, 70% in the bag. So it's naturally going to move from the higher concentration to the lower concentration. Finally, there's those 3 major types of solutions that we talked about. Hypotonic, where we have less solute and more water. So water is going to want to leave hypotonic solutions, in this case, leave the beaker and go into the cell, where it might pop the cell. We have hypertonic solutions, where we have more solute and less water in a hypertonic. So water is going to want to move into a hypertonic. So it's going to leave the cell and go into the beaker. So the cell is going to become shriveled up like a raisin. And then finally, we have our isotonic solution, where it's balanced between the inside and the outside. Same amount of solute and water inside and outside. So the cell is going to remain nice and happy, and the water is not going to become more concentrated on one side or the other. It's all nice and balanced. righty. So like I was saying at the beginning of class today, the assignment for today and also the assignment for tomorrow goes directly along with the notes that we were just looking at. And so you can fill in the information in these blanks right here with the material that you find in the notes. And then Wednesday's assignment is just going to be a simple question about hypotonic, hypertonic, and isotonic solutions. So keep an eye out for that to be posted on Wednesday. All right, so I know that was a lot of review right there at the end. I'm sorry about that. I just realized about halfway through the lecture that I totally forgot to hit the record button. But hopefully that helps out if you want to go back and rewatch me go through some of that material once again.